Good morning, everyone. Let's get started. It's 10:33. Welcome to one more session of our UC Davis Team Research Forum. As we have discussed in the past, we have been making an effort to bring in more of our colleagues and partners from the national labs to learn a little bit more about how to engage with researchers at the labs, and what are some opportunities for specific areas of partnership and collaboration. So today we are hosting our partners from Lawrence Livermore National Lab. We are going to have a little bit of an introduction to the lab by our colleague Amy Greishuk. We've been working closely with Amy over the last year on a couple of engagements, including the lab fees workshop that was focused on pandemic preparedness, wildfires, and just having regular conversations around what are some topic areas where we can assist our researchers in coming together. So I'm going to let Amy go ahead and start her introductory presentation. We just remind all of our participants to please keep your microphones muted so we can reduce the background noise. If you have questions, please go ahead and post them to the chat, and we will get to them at the end of the presentation. Thank you for joining us today, and with that, I'm going to let Amy get started.
Thank you. So just checking my sound, and I think the slides are now presented. All right, so thank you, Ana Lucia. It's a pleasure to be here today. I'm also joined by my colleague, Dr. Bruce Bucholtz. As Ana Lucia mentioned, within my role at Lawrence Livermore National Laboratory, I focus on strategic partnerships. I sit within our discipline organization of the Physical and Life Sciences Directorate, and we have an office focused on strategic science engagements. And my role as an investigator is to work with our researchers to build those collaborations. And that is with other national laboratories, with government agencies, industry, nonprofits, and with academia. So very excited to be here today at the Team Research Forum. And in the next few slides, I'll give an introduction to Lawrence Livermore, and then I will soon hand it over to Bruce. So Lawrence Livermore is one of the United States Department of Energy national laboratories. There are 17 of them, and they are located across the nation. You will see Lawrence Livermore here in Livermore, California. We are close to Lawrence Berkeley National Laboratory and the satellite campus of Sandia National Laboratories with their home base there in New Mexico. And across the map, you also see there are additional specialty laboratories, for example, some accelerator laboratories. Next slide, please. Lawrence Livermore was established in 1952. It is one of the DOE National Nuclear Security Administration laboratories, and we have approximately 8,000 employees to date. And our director is Dr. Kimberly Budil. We sit on 1 square mile in Livermore in the East Bay, and that is about 80 miles from UC Davis, I think, from the medical campus, and it's about 45 minutes outside of San Francisco. So we are east of San Francisco. Our annual budget is approximately $2.8 billion, and we are a GOCO. So we are government owned by the US Department of Energy, and we are contractor operated. For a long while, we were operated just by the University of California. And I think close to the 2009 timeframe, we are now operated by Lawrence Livermore National Security, LLC. And you can see UC California as well as other consortium organizations. Next slide, please. So in our next slide, we are seen as a federally funded research and development center. And we have focused on, apologies here, our mission area is to have a focus in world-class science and engineering. And we focus on the world-class science and engineering for innovation with a global impact. And the national security challenge we're looking to address are areas within biosecurity, cybersecurity, energy and space security, with programmatic mission areas in non-proliferation, counterterrorism, and defense. And in order to tackle these challenges, we're looking at multidisciplinary research, partnering with academia and industry, and really utilizing unique research tools and facilities. And so you'll hear about one of those unique capabilities from Bruce in the next few minutes. Next slide, please. And then I want to end with our Livermore Valley Open Campus, really looking at promoting and building and expanding our collaborations. And one thing I want to mention on the previous slide, we had a careers website, and I'll put it in the chat message box. There are opportunities in which we can look at working together. That can be through student internships, postdocs, and as well as faculty sabbaticals. Those faculty sabbaticals are quite unique. They can be 1 to 3 months. They're fully funded by Lawrence Livermore. So if you're interested in that, please let me know. But within our open campus here, you can see that there's new buildings that we have. And within this 1 square mile, we normally have gates in which you have to have all the permissions in order to access the property. And so in the open campus, they push back the gates and they move it inward so that we have this open space, and it's on the eastern side of our laboratory. And you take a public access road, you can pull in, you can park just like a university setting or industry. And we have some new buildings that opened in August, so new office space and collaboration centers. We also have a standing advanced manufacturing laboratory in a computing complex. And then we have some new refurbishments that sorry, Ana Lucia, just checking there. I think there was some noise. Can you hear me okay?
Yes, we can hear you okay. Apologies for that. With just a reminder to our participants, please keep your microphones muted.
All right, certainly. So, so then to end with the open campus, what I think is really exciting is there are a few buildings on the open space that are currently being renovated. And this is actually space that is leased by the University of California Office of the President, and it's opportunities in which we can build and explore additional collaborations with the UC campuses. So I will do a quick introduction to Bruce and hand it over to him, but please know if anybody is interested in conversations from today or exploring opportunities, please feel free to reach out to me by phone or by email. All right, so next slide, we're going to be hearing from my colleague, Dr. Bruce Bucholtz. He is a senior scientist at the Center for Accelerator Mass Spectrometry at Lawrence Livermore National Laboratory. And he is a project manager for the NIH User Resource for Biological Accelerator Mass Spectrometry, BioAMS. For the past 25 years, he has designed and conducted C14 tracer biological AMS studies with collaborators around the world in the fields of nutrition, environmental toxicology, cancer biology, pesticide ADME, and cell biology. He holds a BA from Ripon College, MS from Northwestern University, and a PhD in nuclear engineering from the University of Illinois. Before joining Lawrence Livermore, he completed postdocs at Argonne National Laboratory in nuclear chemical engineering and at UC Davis in pesticide ADME with Professor Bruce Hammack in the Department of Entomology. Dr. Bucholtz has published more than 100 peer-reviewed papers. Bruce, I'll hand it over to you. Thank you. So I think, Bruce, you may be on mute still.
Sorry, I've had a long association with Davis since I first moved to California 25 years ago. When I first came to the lab, it was as a UC, what was the precursor of the UC fee program, where I was in residence in Livermore, but in Bruce Hammack's lab. So that was the long association. Today, I want to go over what accelerator mass spectrometry is and how you can use it in the biological sciences, and focusing on the BioAMS program here at the lab. I'm going to illustrate AMS capabilities with a few published examples, and then describe how you can access the BioAMS user facility at Lawrence Livermore. Center for Accelerator Mass Spectrometry at Lawrence Livermore was formed around 1990. We're at the forefront of AMS technology, and we work in many different fields. I'm going to concentrate on biomedical studies today, but I list many of them that we work on here at the lab. The picture there is of our large accelerator system. That was the first one that was put in place here. It's a 10 megavolt system that fills a warehouse. It's very impressive, but it isn't as easy to use as some of the newer systems. We have a small compact system that we use for carbon cycle work. Was originally purchased for doing biomedical work, and now we have a dedicated biomedical accelerator system in our biosciences and biotechnology division, which takes up the space of a normal biology lab. So we started with a system that measures everything on the periodic table, fills a warehouse, and our current system fills a normal lab, and it is designed specifically for measuring carbon-14. So in the last 25 years, AMS has evolved into an innovative biomedical tool. We've have data developed for AMS technology that's helped in risk assessment of toxicants, a wide variety of different types of compounds, addressing safety and efficacy considerations of therapeutics, understanding xenobiotic intermediate metabolism, interest in eliminating interactions between critical molecular pathways in various disease models. Done a lot of work strengthening models for metabolic and biological states, examining turnover of long-lived cells, proteins, and then provide quantitative comparisons in metabolisms and dosimetry between different species. AMS measures isotope ratios. So it's an isotope ratio mass spectrometry technique. We have very high selectivity, sensitivity, and precision. How it is, say, we are always measuring a radioisotope here, but we're not treating it as a radioisotope. We use a linear acceleration stage that brings our ions to high energies, and it allows us to count carbon-14 atoms, atom by atom. So we are actually counting atoms rather than measuring radioactive decay. And we're using the carbon-14 like a stable isotope tracer. Over the course of the experiments, there's not a lot of radio decay that happens, and most of the samples that we measure are not technically radioactive by definitions by DOT or hazardous waste and waste regulations. So we're able to measure attomole quantities of agents, metabolites, and adducts in small milligram to microgram sized samples. So the AMS excels in research that requires radio-labeled agents. The studies are ideal that need very high precision, and sensitivity is an issue. Our dynamic range is about 0.6 to 600 femtocuries per milligram carbon. The natural level of carbon-14 in the environment is about 6 femtocuries per milligram. So our dynamic range is really about 1/10 the natural level to a couple of hundred times the natural level. Detection limits are attocuries per milligram carbon, and typically attomoles of agent with 5% precision. The studies of the agents are typically things that can't be produced at high specific activity, where samples are very small. Examples where you have very small samples, you might be working with a compound that is highly toxic or carcinogen, so you need to use a very small amount of material that is not a hazard. We have a wide range of applications, a lot of dose response, mass balance, ADME, which is absorption, distribution, metabolism, and elimination studies, pharmacokinetics, pathway analyses in animal and human subjects, measuring absolute bioavailability, looking at protein and nucleic acid binding for adducts, measuring metabolic parameters, and then cell turnover or protein turnover in humans. How AMS differs from conventional mass spec There are a couple of major differences. One is that we're using a radioisotope tracer instead of a stable tracer. We have higher sensitivity than typical mass spec. We work with solid or liquid samples. Conventional mass spec typically is solid or liquid, but they can also do gas. We are a quantitative technique, so we are measuring numbers, we're measuring concentrations. Conventional MS, sometimes as quantitative, often as qualitative. We do not provide any structural information in the AMS measurement, while conventional MS can provide that. We have unrivaled specificity and sensitivity of our tracers. That's because the only elevated C14 in the sample is coming from the tracer material that we put into the experiment. In conventional mass spec, you do not necessarily know what your metabolites will be. So for AMS, we know if there is C14 that's elevated above the natural level, that's coming from our dosed compound. So we don't need to have a priori knowledge of what our metabolites will be. I mean, ideally, you combine conventional MS and AMS to get both the exquisite quantitation from AMS, but then also structural information and identification with MS. And that's actually where we are moving today. Give you an idea of the range that we work with. The pink box is the range of concentrations of carbon-14 in the biosciences. Often people refer to modern as a concentration. Modern is just the natural level of carbon-14 in the environment, but 1.2 parts per trillion. So 13.5 DPM, disintegrations per minute for a gram of carbon. And we are looking at variations off of this natural level for the materials that we trace. This is a picture of our biosystem currently in the lab. What we're doing, again, we're tracing molecules in the biological system using the rare isotope. So we're counting the atoms. We're comparing a carbon-14 atom count to a stable isotope current. So AMS is about 100,000 times more sensitive than decay counting for carbon-14. So we can measure very low levels. We're following the compound through chemical processes as the elevated carbon-14 comes solely from the starting tracer compound. Give you an idea of the types of samples that we measure. For solid carbon samples, we take a material, we combust it to carbon dioxide and then reduce it to graphite or a solid target. And typical size samples there in the table you see on the left: 12 microliters of plasma, 5 microliters of blood, so a small drop of blood. Urine has a lower carbon content, we need more, and the other materials as well. So this is really ideal if you need to do frequent sampling or high density data, if you're looking at a metabolism study. So you give material, look at how it goes into the blood and then gets cleared. A single drop of blood provides a sample if you're not looking at metabolism. So you can get very high frequency data on the rise and fall of a compound in circulation. Proteins and DNA are about 35% carbon by mass. And if you're looking at adduct formation, typically have to add a carbon carrier because you don't have a 0.5 milligram of DNA or protein in most samples. We use an undosed tissue as our control because we're measuring an elevation of carbon-14 above the natural level. Currently, when we do LC, we're not doing fraction collection and conversion to elemental carbon. In most cases now, we are using a direct interface where we take the LC eluent, put it on a nickel wire, combust it, and inject the carbon dioxide directly into the ion source. So the new way of doing LC is doing it all by carbon dioxide. And the old way of doing tissues, we still use by graphite. So I'm going to again go into a couple of applications now, looking first at cancer research and clinical applications. The very first studies that were done with AMS were looking at food carcinogens. So methyl IQX is one of these carcinogens that's made when you cook meat. Early studies looked at humans and rodent studies of the food mutagen methyl IQX. Let's look first at the HPLC traces on the right. You can see that humans and rats do not metabolize methyl IQX in the same way. You have different dominant metabolites. And humans actually produce more of a bioactive metabolite. So when we looked at binding, excuse me, of methyl IQX in humans and in rats, we find that the levels are actually higher in humans than in rats. So this is where a situation where if you were trying to do your study, so try to determine binding of a compound in humans, the rodent data would not predict it particularly well. Another thing to see here is the spread of the points. In the rodents, it's fairly tight compared to humans. And the reason for that is that rodents are typically very inbred. Their metabolism is very similar. Humans are not inbred. So you get a much wider range of metabolism and binding in humans compared to rodent studies. Another application is looking at trichloroethylene, which binds to DNA. It's widely used industrial chemical, and it's a low-level contaminant in surface and groundwater. Here we looked at binding of TCE to DNA and protein. You can see that we're able to go over 5 orders of magnitude, and the sensitivity was only 5 ADAX per 10 to the 12 nucleotides, so quite low level. This is the thing I'd like to excuse me, look at most closely. AMS is really good for doing microdosing, also known as a phase 0 study. I'm going to just describe a study that was done at UC Davis Cancer Center in the next slide. And microdosing is an approach to deliver a subpharmacological level of a compound or drug to healthy volunteers and then do a full metabolism study. A microdose is 1/100th the level calculated to yield a pharmacological effect, or 100 micrograms, whichever is lower. The idea is that you look at how the compound is metabolized. You're looking for cellular response, see where it's binding, but you're not expecting a whole body effect. So there's not enough material to have the therapeutic impact that you'd expect from the drug. So this is really useful for doing an early in-human study. So either a microtracer, or another approach that's been used by pharma is to put a small amount of C14 in a phase 1 study. And phase 1 is toxicology. So the reason you would do that is that you would look to see, is there a difference in the metabolism of the compound as you increase the dosage? Some compounds can be metabolized by different ways. So do you saturate one pathway and then metabolism is coming through a different pathway, and perhaps you get different metabolites. Perhaps one of those metabolites is reactive. So this is really good to look at drug candidates, and you can lower the risk of adverse effects by having a very low level. But the technique is particularly valuable in cases where the human in vivo performance is difficult to predict, either from in vitro or from animal studies. And for example, look at the methyl IQX. The metabolism and binding was significantly different between humans and rodents. Both the FDA and the European equivalent of the FDA, the EMEA, have accepted the microdosing approach for early first-in-human studies. They allow the companies to make less of compound than typical phase studies, and they have then less expense in getting a compound into humans early. And the advantage here is you get it into humans earlier, you can see whether or not it's going to fail early, and you can save a lot of money in development of a compound that will not make it to clinic. So in the study at Davis looked at platinum-based chemotherapy, and the drug they used was carboplatin here. So it's a platinum stuff, and there's carbon on it, so we could put a C14. In this study, this is for people who were about to that had different types of cancers. The study was done in non-small cell lung cancer and in bladder cancer. People were given a microdose of carboplatin, waited 24 hours, and then either actually did both a blood sample and a small biopsy, and looked at binding of the carboplatin to the DNA. So injection, collection, a day later, then look for binding. If somebody is going to be sensitive to the carboplatin, which is basically forms adducts and it prevents cells from reproducing, you get a bunch of adducts formed. So that person would be sensitive, or that cancer would be sensitive. If you don't have adduct formation, it's not sensitive, it's resistant. So by looking at the amount of binding of the material of the drug to DNA, you may have a way of being able to tell whether a particular cancer is going to be sensitive or resistant to chemotherapy. And why is that important? Chemotherapy is very, very hard on the body. And if someone is not going to respond to using a particular drug, it'd be better not to use that and put them through basically the insult of chemotherapy, because in many cases there's more than one drug available. So how they measured the difference is a responder had a larger shrinkage in tumor compared to a non-responder. So some data from the study. This PBMC is blood, peripheral blood cells, looking at carboplatin monoadducts. Responder is the circles up here. Non-responder is square. You see, it's not universal. The responders on average had a higher binding, but there were also some that were lower. Tumor actually seemed to be less productive than the blood. And it's not entirely clear why, but part of it could be that the blood circulation is not necessarily great in all parts of the tumor. So the actual exposure of the carboplatin may not have been uniform in the tumor. So in tumor was not nearly as good. Here we could be able to look at the levels of binding of the platinum to DNA damage may be predictive of chemotherapy resistance. So in this case, all the subjects went through full chemotherapy of the drug. We didn't know whether they would be a responder or not a responder, and they were actually looking at tumor shrinkage as an endpoint. The pharmacokinetics of the therapeutic dose and the microdose were very similar. So that's why they picked 24 hours as a time point for doing this. They've also done studies looking at xenograft tumors in mice and looked at more than one compound. That study becomes very complicated to explain. So decided not to present that right here, but that's also available. Another study that we've done looking at cancer is trying to measure metastatic potential of cancer. So we grew this is prostate cancer lung cells, labeled it with thymidine at a very high level, and then injected the cells into a mouse and looked to see where the cancer cells ended up. So we were able to, excuse me, look like weeks later to see whether a tumor was formed. It was very, very low level of of these cancer cells actually stuck in any one place, but we were able to look at quite low levels. A number of labels per tumor were actually, we could actually measure quite low levels. And I think we got down to being able to measure a couple of tumor cells in a sample. Let's look at biomarker identification and environmental contaminants. So this is an early study that I was involved in. It was a dermal application of the herbicide atrazine. Atrazine is pictured up here. It's a triazine. So it's a carbon nitrogen ring, and you have different groups off of it. When atrazine is metabolized, the chlorine can come off, and you can get different groups on these parts. So it's quite complicated metabolism. So the study was done in humans based on an animal dosing, and I came to this study after the human study had been done. The amount of C14 in the urine that was collected was sufficient to measure atrazine levels, but not enough to do metabolism using a decay counting method. So what we did was separate urine samples and then measured fractions with AMS. What we found is that the atrazine mercaptan was the best biomarker of dermal exposure, and that's this peak right here at about 39 minutes. These different colored traces are at different time points. The red is 4 to 8 hours after the start of the exposure. Blue is day 2, yellow is day 7. And this is normalized to the total amount, excuse me, of material. What we found is that over time, the metabolites became more polar, which were earlier in the elution here, and the total amount also reduced. There was no parent compound found in any of them. So what does this tell you? One is that if you're trying to determine an assay for an exposure, when you're collecting the sample is important, because you're going to get a different metabolite profile, whether you're sampling shortly after the exposure, 4 to 8 hours, or 7 days. You're going to get different compounds. So you're going to have a different assay depending on when you sample. And important thing here, no parent compound was found in any urine sample. So there was actually quite a bit of stuff in the literature talking about the measuring parent compound, and we know that there wasn't any parent compound if something was actually metabolized. Okay, this is another study that was done very similar to the atrazine study, looking at the pesticide permethrin. Permethrin is very widely used to control insect pests. You find it in a lot of home and garden products. It's used on mosquito nets in malaria areas. You'll find it on clothing. The military uses it to reduce insect vectors on troops out in the field. You can buy hunting clothing, fishing clothing, golf clothing that is soaked in permethrin to reduce mosquito exposure. So in this study, we used amount of permethrin that you'd find in a lice treatment. It's also used to treat lice. It's painted on the forearm for 8 hours. We put the label in 2 different places, because when permethrin is metabolized, it breaks right here, and you have two different types of metabolites. We collected saliva, blood, and urine for a week. Average absorption was quite low, only about 2.2%. Maximum serum and urinary excretion rates were 6 to 12 hours after start of exposure. So here's a trace of the metabolites that we saw. So most of the subjects in the study had a mixture of these 2 labels, about 50-50, because we wanted to see what's totally formed. One subject had only the phenoxy label, another one only had the cyclopropane. We see so that we can distinguish them. Turns out that most of the C14 was in polar peaks early, before we had any standards. This study was done quite a while ago. These are all fraction collection. So this was really quite cumbersome to do this study. 3-phenoxybenzyl alcohol glucuronide was the most abundant metabolite identified on the things that we had. Analysis of urine samples using an amino assay for 3-phenoxybenzoic acid, or 3-PBA, showed that acid hydrolysis result in the appearance of 3-PBA. So we believe that these are probably some of these metabolites of 3-PBA, and that is the biomarker that is used. So these two examples are the old way of doing an LC. We are currently doing parallel accelerator mass spectrometry to do LC analyses. How we do that, instead of collecting traces, we can put the eluent directly on a wire and use CO2 as the injection and do the quantitation directly. So this system, we have a UPLC up front, we have a flow splitter that can go either 100% AMS or 100% MS, or any increment in between. With AMS, we combust the CO2, we inject that CO2 into the AMS, we get quantitation right here. We get C14 peaks, and we have carbon peaks. So you can see right here we have the gray, there's zeptomoles of C14. So these are labeled materials. With the MS, we can get identification. So this peak here is run there, and it's measuring glutamic acid. So this is really a time saver. Doing fraction collection and doing all the sample targets as solids takes a long time. In this way, the analysis is as fast as running an LC trace. So in the old way of doing it, it would take several days to prep the samples, and in one day we could measure one trace. Here we can measure at least 10 times as many traces in the same amount of time. Okay, this is another, I'm going to just pop right through this one and look at ADME and health and disease. We did a study looking at aflatoxin in peanut butter. Aflatoxin is a potent toxin that's found produced by foods. It's found in corn, cottonseed, peanuts, and milk. We're able to give a dose of humans the amount of aflatoxin you get in a peanut butter sandwich. There's no real health worry that people have. We're able to measure the C14 in blood and urine and see that eating chlorophyll or chlorophyllin along with the peanut butter or along with the aflatoxin actually reduced the amount of absorbance. In this study, the amount of C14 was 5 nanocuries. People were able to do all three of these. So these are average traces, but the same person could do each treatment. The amount of C14 was so low that we didn't have carryover issues. This is a study done with Andy Clifford, who's since retired at Davis, looking at folic acid incorporation. Folate you find in leafy vegetables, and it's currently being used to fortify flour as a way to reduce the amount of people who are deficient in C14 in folic acid. So this trace here is, I believe, 10 subjects in plasma and then in red blood cells. You can see here that we could follow this out for 200 days. The dose was 1/12 the recommended daily allotment, so quite low. Andy was able to construct a model of metabolism and show that by increasing the amount of folate in diet, you can deal with basically eliminating birth defects, which is why the flour is now fortified with folate, because many people were deficient in folate. Now, if you eat flour products in the US, you are not folate deficient. And hepatitis is a problem also. People who have reduced liver function have a problem, are often folate deficient. By increasing the amount of folate in the diet, we're able to reduce that. Another project we worked with is a vitamin B12 deficiency. The trace here are different subjects. PA1 is someone who has pernicious anemia, which is inability to absorb B12. Here we labeled vitamin B12, which was actually quite difficult. It's a large molecule. Give it to people and then measure the absorbance. So a microdose of C14B12 could make an easy test to determine whether or not someone is an absorber or not an absorber. So B12 deficiency is really the cause of pernicious anemia. It's very common in the elderly, but this is a way you could actually tell whether or not somebody is able to absorb or not. You can give them a small dose of B12, sample about 8 hours later or 12 hours later, and then you can see what the level of C14 B12 is in their blood. You could tell whether or not they are absorbing B12 or not. So we've also looked at flour as a means of putting the B12 in, and then also in eggs. Those are two different studies. We found that putting the B12 with flour, basically putting in bread, had the same absorbance as the crystalline, like little pill, and with eggs as well, you had the same amount of absorption. And then do a quick look at another type of study that I'm involved in. There's something called the radiocarbon bomb pulse, which was a result of the US and the Soviet Union doing above ground nuclear testing from the late 50s until 1963, which was the nuclear test ban treaty. What those above ground tests did was double the amount of carbon-14 in the atmospheric CO2. Atmospheric CO2 is incorporated into plants and into animals, and basically the atmospheric testing labeled the entire biosphere of the planet. What we did with collaborators at the Karolinska Institute is develop a technique to isolate DNA and measure the carbon-14 content in DNA to determine the age of cells. And here this is the first paper we published that made the cover of Cell, able to show that the C14 content in the DNA of cortical neurons matched up with the time of birth, which means that you don't get any new cortical neurons. We've looked at other types of cells as well. Hippocampus does turn over time. Hippocampus is the area of the brain where memories are converted to long-term storage. We've looked at heart cells, and so that the turnover of heart cells is about 1% annually. So over your lifetime, you actually replace about half of your heart. We've looked at long-lived proteins as well, proteins in the eye, collagen in blood vessels and in aneurysms. So this is a technique that we can look at turnover in health and disease in humans by getting autopsy samples and excuse me, develop models and understand what natural levels of turnover are of a great number of types of cells. This information really wasn't known before we started doing these studies, and it's been very, very helpful. I'm going to just go over key points here. AMS measures extremely low concentrations of radioisotopes, and it can be used in a wide variety of studies. It's most helpful in looking at pharmacokinetics, anatomy studies in humans. We can remove any obstacles of radioactivity concerns because we can work with amounts so small that they're essentially not radioactive. We can facilitate relevant studies in metabolic pathways and health and human and human disease with lower and biologically relevant concentrations. We don't have to go to high levels to measure. We can measure natural or environmental exposures of a wide variety of compounds. I want to get to how you can access us. CAMS is the home of the National User Resource of Biological AMS. We're an NIH center. We're the only center of this type. We have over 30 years of experience in developing and applying AMS in biomedical sciences. The technology is available to all biomedical researchers. In most cases, the measurements are free. There are limits if you have too many samples that you're going to have to pay for some of them, but it is quite easy to fill out an application, download it from our website, and apply to use the resource. Applications are reviewed continuously by the resource staff, so there's no deadline. The website there is bioams.llnl.gov. I'm the point of contact on the website. You can email me directly. I believe my phone number is there as well. And I'd be happy right here to take any questions.
I think Ralph has a question. Go ahead, Ralph.
I do. Thanks, Anna. Bruce, that was a lovely presentation. I have a couple of questions relating to well, first of all, relating to the work on carboplatin, where you found a discrepancy between the distribution of those two adducts in the molecule in the tumor itself, compared with the whole patient. Have you thought of perfusing the area of the tumor? In other words, directly administering the C14 labeled carboplatin into the tumor and then measuring what comes out on the venous side?
That was not done. I wasn't involved with the human study part of that.
Okay.
Did measurements. I think they were looking for something that was least invasive.
Right.
So they just did an injection and did it that way.
Right. I mean, you might be able
you're
to do
suggesting it, yeah, it would be better because you would definitely get the carboplatin to the target.
Exactly. Yeah. Yeah. Maybe in a xenograft model, you could do that. I see the problems in patients. Jumping to a different question in terms of a potential application, using the C14 labeled thymidine method, is there enough C14 there to enable radio autography to be carried out on the tissues? So I guess you could measure C14 in the whole tissue extract as a measure of how much thymidine was taken up. But would you be able to, is there enough sensitivity to be able to actually show which cell types have taken up the thymidine?
I don't know. I kind of glossed over that example, and that one, the labeling was actually, the study that was done was actually labeling the tumor ex vivo,
right,
expanding it, then putting it, well, actually they were done a couple of different ways. One was just injecting labeled tumor cells and looking to see where they went, see if they stuck any place. Another way to do it is to implant the tumor in the mouse, then inject the thymidine, and then look to see where it goes. I don't think any attempt has been done to try to separate the different types of cells in the tumor. I think that could be done. You'd have to have probably a fax method for looking for different surface proteins. That's not my expertise.
Okay, fair enough.
But in theory, I think that could be done.
Offline, we could talk about a potential application. If you can get specificity to identify which cells are taking up the thymidine and in what amount. Thank you.
Yeah, and I'd say in theory, you should be able to do it. In practice, I don't know how hard it would be.
Got it. Thanks.
Thank you, Ralph. Any other questions from our audience?
So I have a quick question.
Go ahead, Angela.
Hi, I just wanted to make sure the sample preparation that you need for the tissues is minimal, right? For any tissue or anything that you want to analyze.
Yeah, well, it depends on what you're trying to measure. If you're trying to measure metabolites, things get more complicated. If you're just trying to look at how much of a compound gets into, say, liver versus muscle, then all you need to do is get a small sample of that tissue, and we combust it to CO2 and reduce it to elemental carbon. And that process is not particularly difficult. We have visiting students come into the labs, so it's possible to come down and work in the labs. I have students from Davis that come down. With the pandemic, we haven't had people coming in for the last 2 years very much, but it's certainly possible to do. In years past, there was a shuttle van that ran between Davis and Livermore, which was wonderful for students. They could get up early, jump on the van in Davis, come down to Livermore for the day, work in the lab, get back in the van at 4 o'clock and go back to Davis. That has been discontinued. But I don't know if it's possible for something like that to come back again. But I mean, it's certainly possible to have students come in.
Thank you.
And we prefer to have students come in and process their own samples, actually, because then they have a better appreciation for what they're doing.
Any other questions from the audience?
Ana Lucia, I would just like to mention as well, kind of coming from being a colleague of Bruce, that if there are any additional questions that maybe are easier to address by email, please feel free. I put in the chat window Bruce's email and phone number. I'm also happy to serve as an additional point of contact. Should there be some side conversations. The goal from a session like this is, one, to provide visibility on Lawrence Livermore. We're really right around the corner. So where can we look at, partnering, collaborating? We also, running samples is great, but running samples that can be pilot data that could go into a proposal. As a national laboratory, we can participate in proposal submissions. So NIH, DOD, I know sometimes even universities look at NSF proposals. Livermore can't lead those, but on occasion, if it's appropriate and eligible, we can serve as a sub. So it's just really getting the conversation started today. So we can take conversations offline. And like Bruce mentioned, part of my role is to help arrange if there are external visitors that would like to come on site and visit the laboratory. As Bruce mentioned, sometimes you can come and you can see the facility, and or you might want to bring your students or postdocs. We can try to arrange that, certainly getting all the permissions with the state we're in right now with COVID, but we can look to make those arrangements if interested.
That is good to know, Amy. Thank you. If there are no other questions, I just want to thank our speakers, both Amy and Bruce, for the contribution to our forum, and remind everyone that we are meeting again in a couple of Fridays, and we'll have a presentation around federal funding opportunities from our government relations team at UC Davis and at the UC Washington office. And we'll send that information to everyone later today. Thank you, everyone. Have a wonderful weekend, and we will see you in a couple of weeks.